So when we went to take a look at the arc port, okay, the arc port was that delta that was sitting just basically south of Bradfield. Okay, so Bradfield's sort of sitting here. The delta that we're interested in is this delta right here. And so this is Pine Tree going into, so this is 366 here. So we came up this, and there's a cemetery right here. We took that turn and we parked right here, and our hole, the pit, was right there. Okay, so if you go to the actual the survey site, the map that shows the survey maps in the not Web Soil Survey, in the Cornell the Cornell Soil Survey that's on the web. Okay, that's this road complex right here. So here's 366 going out. Here's Pine Tree coming in. We came up here. Here's the cemetery. Come in through here. It's this garden, basically where the A is in the garden. Okay, so that map is basically C1. Okay, if you go to page C1, you'll see the soil map. Okay, and there'll be a map unit right about here that will be called, it'll be called AKB. AK being arc port, B being the slope. Okay, that's the soil that you're interested in. Okay, does that help a little bit? Okay, Tompkins Soil Survey, comes to my attention that the Tompkins Soil Survey, this is the Cornell Soil Survey. The Tompkins Soil Survey, the library still has not gotten it up. I've actually talked to them a couple times about this. There are some hard copies of it at the library, so go visit them. I also have a boatload of hard copies. So if you can't get a hard copy of the Tompkins County Soil Survey, come see me. Okay? Yes? We're good with that? All right. Next subject is lab this week. Two points about lab this week. We are going out this week, obviously. We're going to go to the top of Mount Pleasant. And it is actually fairly pleasant up there. Remember to wear the correct type of shoes. I know you guys are going to be like, crazy, stop doing that. Please wear the right type of shoes. Okay, but we are going to be leaving. We're going to be meeting here. We're going to be going out to the field almost immediately. We're going to talk about two things in the lab before we go, and then we're going to go straight out to the field. We get out there, we're actually going to be looking at two different pits, and we're going to be talking about some stuff when we get up there. Okay, you'll also get to visit my favorite apple tree. Okay. Last week, I already did the location thing, so everybody's sort of cool with that. If you have any questions about the location, come talk to me. Okay? We good? All right, no questions. All right, so last lecture, we were basically talking about concept mapping and soil mapping, right? So here was the concept. This is actually the concept of the landscape that you're going to be meeting this week. This is the top of Mount Pleasant. Okay? The Martins tend to be at the top, moving down into a shallower bedrocks, shales and sandstones. We get into the Lordstons. It opens up a little bit. We have a concave landscape. We're starting to look at Erie. Okay? Then Langford down in Bath and Valois, Phelps and Howard down in this gravelly area. Okay? Now let me ask a couple questions about this. Just sort of think about this landscape. Okay? Remember this. Now let's go back to this landscape. It starts raining. Okay, and it's going to rain uniformly across the entire landscape. Where's the water going to get concentrated, though? Down at the bottom, right? That makes sense. Yeah, we're going to have infiltration all through here, but some of this is going to turn into overland flow, and so we're going to have a concentration of water down here. Okay, so not only do we have a topographic shift, we also have a water shift. Okay, now let's take a look at up here. Pretty flat up here, and you'll get up there, and it's fairly flat. It's going to rain. Most of the water is actually going to go in, but we're going to get some overland flow. We get into this Lordston. We got a slope. Okay, water, some of it's going to go in, but most of it's going to go sliding off, which then brings us down to this Erie. Okay, the Erie, we've got this concavity. Okay, so we sort of have a hollow in this landscape. It turns out that Erie is going to behave a lot like the soils down here. Okay, this is not a smooth slope where it's just going to go straight down here and then settle out. So if I'm going to start having water, increased water here, what do you think that's going to do to something like this? We started talking about drainage, right? Okay, well, I have a concavity It's being fed water from above, not a lot of water from here, but a good amount of water from here. The water is going to slow down. I'm going to see an increase in infiltration, but I'm still going to have some come off. But what is going to happen to my drainage areas in there? What's the water table going to do? It's going to what? It's going to start rising. Okay, and if it starts rising relative to the well-drained margins, or the moderately well-drained margins, what's going to happen to the drainage? If that water starts rising, those modeling, those features that we're going to see in that soil is going to come higher. Does that make sense? Then what happens down here? Oops, wrong way. We start going back down here. We start getting that slope again. Deeper soils. Any thoughts? What's going to happen to that drainage? What's going to happen to the water table? Let's call it that. It's going to start going down again until it gets down to about here. And so potentially we're going to have higher drainage features or redox features here than we would here. And certainly higher drainage features here than we would see here. We also have this little volusia up here, which is a hollow on the top. Basically, it's going to concentrate all that water down into the hollow. Does that make sense? So you can sort of predict, just by looking at this concept, you can sort of predict where the drainage or the features of the water in the soil are going to be, just by looking. And that's something that we're going to be looking at this week. When we get up there, we're going to be taking a look at margin soils, sitting right at the top. We're also going to be looking at some soils right down at the bottom. Okay, and we're going to see this huge juxtaposition between the drainage. Okay, so interesting, when we talked about this mapping, we put all these sort of features, this concept together, and we start being able to arrange soils in space. In this case, this is an association map, but basically similar properties across this space. Okay, so once we start putting soils similarly together, we can sort of see similar things that are producing these soils. In these cases, mostly due to parent material. We've got the acid soils in the higher elevations to the south. We've got the limier soils, the sweeter soils to the north and in the valleys. Okay? So we can start seeing those trends. We can start ranging these soils based on how they're made. Okay? Basically, we can start classifying them. Does that make sense? So today's lecture, we're going to be basically talking about classification. It's going to be soil classification, how we, in fact, classify these soils. And when we classify, we're basically putting these soils information, we're organizing the information of these soils so that we can basically do two things. One is that the information is understandable for everybody. And two is, not only is it understandable, it's usable for everybody. Now when we classify, we basically classify in two different ways. We have what we called an observed property, or we infer properties. Observed properties we basically call a natural classification system, where we classify based on what we see at that location. Okay? A dog has 4 legs. We see 4 legs. We don't infer what they do with those 4 legs, but we see they have 4 legs. Okay? We can also infer based on those 4 legs, or whatever we actually see in the soil, or whatever we're in fact classifying. Okay? The observed ones, where you're looking at the observed properties, we call that a natural classification system. The inferred one, we basically call that a technical classification. Soil taxonomy is a natural classification. An observed or a technical classification is basically inferred potential, a yield potential, for example. Okay, does that make sense? Natural versus this inferred or technical classification. Now, natural classifications work fairly simply. We're basically looking at phylogeny. Phylogeny is the relationship of different properties over time. Okay? How things are related. Okay? Think of this in a biological sense, how one organism is related to another. Okay? How those characteristics came to be. Okay? We basically put that into a taxonomy, which is the classification of how those things came to be. That's the relationships between them. Okay? Basically, we're looking at things like genotype and phenotype, the differences there. Genotype, basically, we're looking at a conserved biological feature, such as DNA, okay, for us, for basically the most advanced way we do classification with genetics, we actually use mRNA. It's a matrilineal line RNA. It's very well conserved. We see it across all the organisms. Okay? We also have phenotype. So we can look at the use of structural and functional abilities to quantify evolutionary relationships. Okay? So we can look at those 4 legs. Okay? Just because you have 4 legs and this person over here has 4 legs does not necessarily mean you're evolutionary linked directly. In fact, those 4 legs could be totally independently evolutionary result. Let me say this directly. How do I say this? Those 4 legs could have arrived totally independent. One does not necessarily have to come from the other. Okay? So we have the genotype versus the phenotype. Okay? Classification based on the relationships of how something came to be. We classify it based on that. Okay? Taxonomy. Okay, we're going to be talking about soil taxonomy. So we're looking at those soil individuals, how they're related by the properties of those individual pedons. Okay? Observable, not inferred. Okay? So the number, the kind, the arrangement of the horizons, the color, texture, structure, pH, all of those things that we can see. How many legs, ears, hair, eyes, fingers, internal organs. Does that make sense? In soil taxonomy, I threw this over here so you guys could have something to compare to, but this is the biological classification. We start with phylum all the way down to species. Well, with soil, we basically do the same thing. Order all the way down to phase. Okay? Phase is really soil mapping. Series is is basically the lowest end of soil taxonomy. Okay, so order, suborder, great group, subgroup, family, series. Okay? All the soils in the world need to fit, just like every organism in the world actually needs to fit into this classification. If an individual soil or an individual organism does not fit into taxonomy, the system doesn't work. You have to be able to classify everything. Okay. Soil series is the most detailed category in soil taxonomy. There are presently, actually this number is wrong, there's presently over 24,000 different soil series recognized in the US. That's not worldwide. Okay? The soil phase is the most detailed characteristic in soil survey. That's the mapping component. Okay, this is sort of a triangle, a pyramid scheme of what? Pyramid scheme. Yeah, it's a big pyramid scheme. No, it's a big pyramid that basically whatever. It's basically representing the different levels of soil taxonomy. So we have 12 orders. And we'll be introducing you to these 12 orders over the next 3 lectures. These soils, all these characteristics that we are looking at, are based on the 5 soil forming factors, parent materials, so on and so forth, all the way to time, and the 4 soil forming processes acting upon those factors. Every feature that you see in soil is the result of those factors and these processes acting on them. Now there are 12 different, and I don't know if you guys can actually see this. Can you see these words up here closely? Sort of like, you should have it, it was definitely in the PDF. These are the 12 orders, and we're going to go through them so you have an idea about where these soils are coming from. And we're going to talk about sort of the major characteristic. What is the feature that sets these soils apart? Why do we call it this? And why is it set aside from all the other soils? And the first one up there is alpha soils. Okay? Alpha soils are basically soils that have an accumulation of clays. Specific kind of clay, a high activity or a medium activity clay. A clay that has high base saturation, has a lot of nutrients. It's a young clay. It's a soil that has accumulation of clay. The next one down is andisoils. Andisoils comes from the term volcanic, or black. It's Japanese. It basically means black soils. It is a volcanic soil. So the diagnostic feature, the major characteristic of this soil is that it's volcanic ejecta, or dominated by volcanic materials. Okay? Now think about this. If I have a volcanic event and that material comes up to the surface, what's going to happen to the lava that's in that material? it comes up, the lava hits the surface, what happens to it? It cools really, really quickly. And if it cools really quickly, the material, the ingredients in that material are not going to have a chance to form regular structural minerals. Right? Does that make sense? Because it cools like that. Okay? These soils weather very, very quickly, and generally they're fairly fertile. Okay? The diagnostic for these soils is that volcanic material, because these soils form very, very quickly compared to many of these other soils. That's the characteristic that sets these soils apart. The next one down is arid soils from arid. You can sort of guess. These are basically desert soils. These are set aside. These are set apart from all the other ones because of their climate. Next one down is entasoil. Basically, the way to remember this is recent. These are soils that have very little development. These are the fresh deposits of stuff. I have just deposited a pile of stuff from a flood event or a landslide or something like that. It inherits the characteristics of where it came from, but those characteristics are not representative of what's going on in that site. It's a recent deposit. Next one down is the gelosoils. Terms comes from gelid, which means cold, very cold. Okay? Basically, these are permafrost soils or soils with cryoturbation. Cryoturbation, cryo, cold turning. When things get frozen, what happens to them? They expand, right? Okay? And if I have a lot of pressure coming sideways and then things start expanding, what direction are they going to expand? They're going to pop things. This is basically the same the feature that you see as growing rocks in New England. All they grow in New England is rocks, right? No, they grow lots of other stuff. But every time it gets cold, what happens to those rocks in the soil? Those rocks start being pushed up every frost, okay? Well, cryoturbation is basically the mixing of material due to frost. Okay, frost, it freezes and then it thaws, freezes and thaws. Okay, and the next one down is histosoils, another Greek term. Histos means tissue. These are organic soils. The thing that sets these soils apart from all others is tissue, organic matter. These are organic soils. Next one down is inceptisoils, inception. Okay, these are the soils that basically come from entasoils, but they're showing signs of development in place. They have moved through, they've basically reached the first stage of maturity. They're moving towards a more mature soil. Next one down is mole soils. Mole is for soft. Mole soils, these are the grassland soils. The feature that sets these guys apart are grasses. The grasses are the formative element of these soils. Yes, parent material is a big part, but these soils are formed under grasses. High bases, good amount of organic matter, and that organic matter is deep. Okay? The delivery of organic matter is not leaf fall, but grasses dying and leaf and roots shedding. So these soils tend to be very dark, deep organic soils versus what we're going to see today, where we see organic, or this week, where we see organic matter basically concentrated at the surface. Next one's down is the oxasoils from oxide. These are very old soils. They have oxic horizons. They have no argillic. The clay accumulation is gone. Okay? Basically, we're looking at highly weathered soils. Lots of clay, but the clay has basically been transformed in place. It's not a translocation of clay like we've seen at the arc port. Okay? Where the silt got weathered, the clay moved down. That's not what these soils are about. These soils are basically taking that material. They've been there for so long that the material has basically been weathered until all that's left is clays. Okay? Next one down is the spodosoil, spodosoil. Okay? This is the soil that I showed you a couple pictures of to show you the E horizon. The term where this came from, it's another Greek. Spodos means wood ash. Remember me telling you the story about these spodosoils when they first started seeing them? They thought they were forest fires. Okay? Is yes? No? Kind of? Ish? Okay. Let me stop for a second. I'm going to show lots and lots of pictures of these things, just so don't get frightened of me just going through these lists. Okay? I just want to give you an idea about how these things are set aside. Okay? Spodosoils. They're due to the spodic epipedon, not spodic epipedon, the spodic horizon. But basically, we're looking at concentrations of iron and aluminum oxides, as well as humus accumulations, organic accumulations. Okay? These are cool boreal soils generally, though there are some in Florida, for example. Okay? They're generally found on coarse, sandy type of parent materials where there's plenty of leaching that can occur. Okay? With acid organic material as it decomposes. Okay, next one down is an alta soil. These are very similar to the alpha soils where we're seeing accumulations of clays. The alpha soils, though, have high base saturation. They're young clays. The alta soils, on the other hand, have low base saturation. Okay, base saturation basically means how many bases are in that system. Okay, so in this scenario, we're still having clay translocating down lower in the horizons. But in this case, the clay is low activity, low bases. Okay, this is the type of soil that you see in the southeast. The alpha soils, those are the soils that you see in the northeast and the upper elevations of the Great Plains. Very fertile. Okay, the last soil is a vertisoil. Verto meaning turn. Okay, this is very similar in the turning nature as the jello soil, but in this case, rather than ice, we're looking at a particular kind of clay that's an expansive clay. It's a kind of clay that if you wet, it will expand. And if it gets wet and it expands, it's going to do the same thing as ice when it expands. It's basically going to rupture, push soil up, and then when it dries up, it will shrink again and start collapsing. And you basically have sort of a clay turbulation versus a gelic turbulation, ice turbulation. Does that make sense? Yeah? Okay, so that's just a brief introduction to these the 12 orders. Okay? Example is the arc port that you've already met and the martin that you're going to meet today. But the formative element is alpha. Arc port is an alpha soil. Martin is ept. It's an incepta soil. Okay? These terms and how we read these, you guys are going to learn over the next couple weeks. Okay? If you get an opportunity in the next couple days, I'd like you to go out in the hall and take a look at the poster that's about soil taxonomy. I will be asking you to do it on Friday, but you can do it earlier if you want to, just to get a sort of a head start on what we talk about here. Okay, we good so far? All right. So I've introduced you to these soils. Let's actually talk about how they are in fact related to each other. Okay, and generally we sort of just show this by basically a sort of a temporal trend here, a longitudinal study. We start over here. This is very slight weathering. Over here, strong weathering. So basically what you're looking at is a degree of intensity or soil weathering. Okay, so degree of weathering and soil development. Okay, so let's start on this side. Okay, very slight weathering. We basically have the entasols, recent. Okay, this is the fresh deposit. Okay, now there is a break right here between this soil and that one soil at the top because all of these 11 that are all tied with this arrow, these are all mineral soils. The one that's on top is the histosoil. Those are organic soils. Okay, or basically with organic soils, you're basically looking at a deep accumulation of organic material. It doesn't talk about what kind, it's just a deep accumulation of organic material. Okay. So let's talk about the mineral soils. The very first soil, i.e. the first deposit, what happens to this material, or how do we classify it? We call it an entasoil. Now, if I have weathering occurring to this soil, depending upon what kind of weathering, if it is mild weathering on volcanic material, we call it an andasoil. So that first volcanic event that deposits everything, it's still an entasoil until it starts weathering into the andasoil. Okay? On the other hand, if I have this recent deposit of material and it's in a cold area where I might have permafrost, and I have any kind of cryoturbation or permafrost, we call it a gellosoil. The frost is producing a diagnostic feature of that soil from the entasoil. It's now has diagnostic features. It's got 4 legs or ears or whatever it happens to be. It's now a gellosoil. If I have mild weathering in a sort of in various conditions, not volcanic material and not cold, we move from an entasoil into an inceptisoil. I'm seeing the beginnings of soil horizons. We have more weathering. We start seeing more and more weathering. And I start seeing clay accumulations. I start seeing alpha soils. If I have more and more and more weathering, I start seeing ultisoils. Entisoils through inceptisoils. From inceptisoils, we go to alpha soils. We started having accumulations of high activity clays. The weathering continues. Those clays start weathering out, and we start getting to low activity clays. And we get ultisoils. Does this make sense so far? OK. I'll take that as a no. No, really. Are we starting to see the trend here? All right. Now, if we start with our entisoils, and the weathering produces a specific kind of clay that has this shrink swell capacity, high base saturation soils with high activity clays, we basically have vertisoils. Expansion, contraction, expansion, contraction. Okay? I will be showing pictures of this so you can see these soils. These soils tend to be dark just because of the nature of the clay, okay, and the organic matter that's in them. Okay. If this entisoil is subjected to a grassland climate where I have lots of grasses, so it's semi-arid to arid, but it's still got a little bit of moisture in there, I start having grasses. Those grasses dominate the soil formation and basically produce mollisols. If instead of dry environment, I get a cool, wet environment, and I have sandy parent materials, I start forming spodosoils. And in time, all of these soils, if the climate and the landscape is fairly stable, sooner or later, all of these guys are going to become oxysoils. Now, the way this flowchart spreads out, it makes it look like all of these go to end states. It's going to get here, and it's going to be a mollisoil, and it's going to be a mollisoil forever. The reality is, all of these soils, including the oxysoil, can undergo some sort of disturbance and go right back to entisoil. Or any one of these soils can go under some kind of disturbance and actually move from iridosoils into alpha soils, or iridosoils into mollisoils, or mollisoils into alpha soils. Okay? This is not a flowchart to the end. Okay? You can start here, you generally end there, but all of these spots, something can happen to them. The climate can change, some sort of stochastic event, a landslide or something like that can happen and basically start this cycle all over. Does that make sense? Yeah? Thank you for the response. Go.
But for the inceptisoils, is inceptisol basically like phase 2 of soil formation if entisol is the first?
Yeah, I mean, the question is inceptisol basically sort of phase 2 if entisol is the first? In a sense, you can think of that. And in fact, most of these soils, the first thing you're going to see is inceptisol. Okay, yeah, it may phase fairly quickly into an andisol or vertisol or gelosol or something like that. But usually this is the incipient conditions. Okay, this is where you see those BW horizons. Remember, we've all been introduced to BW. BW, weak color or structural development. Okay, we saw it right underneath that AP horizon in the arc port. Okay, you're going to see it very pronounced this week. Feel comfortable with this? Now, this is the flow chart of weathering and development and soil development. This is sort of a box chart looking at sort of climate versus temperature. So moisture versus temperature. We're basically looking at climate. Okay? And the way to think about this is imagine this slide right down here. What is actually driving this? This is not a timeline. Okay, we sort of portray it as a timeline, but this is really intensity of weathering. Okay, I could have an environment that's been there for 1 million years, but no weathering occurred. Think of a dry desert environment. On the other hand, I could have a climate that dramatic moisture, hot temperatures, weathering is going to be a lot faster. Does that make sense? So in that scenario, I'm going to be driving the system that way a lot faster than I would be in the desert. Okay? So let's actually look at what the climate does to where these soils are. So let's imagine a very intense weathering environment. I would imagine a warmer temperature, maybe even hot, certainly a moist, perhaps not drowning underwater, but a moist environment. So if we think about that, warmer and wetter, what do we end up with? Those oxasols, right? On the other hand, let's put the scenario where I have an environment, very poor weathering environment. So cold, dry environment. Okay? Not a lot of stuff is going to be happening there. Cold, dry. We've got gellisols. EIH basically means entasols, inceptisols, and histosols. We're looking right in here. Does that make sense? Yeah? Now, if it's a cold environment, it doesn't matter if it's wet or dry. Okay? I'm still going to get those frost, that frosted environment. Okay? As it gets hotter and dry, I'm going to move right into these antisols. Okay? As it gets hotter and wet, I'm going to start moving in. Basically, if you follow this trend right here, I'm going to go straight through alphasols into ultrasols and ultimately to oxasols. Does that make sense, that arrangement? In essence, we're just basically doing a pseudo degree of weathering and soil development. Go.
So the vertisols are spirisols are like that anywhere in that area?
Sort of, we find them sort of in here.
So how do you get that versus getting like an alphasol?
Well, vertisols so the question is, how do we get the vertisols versus alphasols or ultrasols? Vertisols are clay soils, but they're a very specific kind of clay.
So it's just if that clay happens to be there.
This happens to have a lot to do with the parent material, but you also have to have the climate to produce those types of clays. And they tend to be centered right in this zone. Does that make sense?
Yeah.
Okay. Sounds good? Vertisols are interesting. Generally, we think of these sort of down in Texas and that area. But actually, we have a lot of vertisols in western New York. There's a whole development that got put on these vertisols. And basically, all the basements are being ripped apart, or were being ripped apart. Imagine you put a material on something that's going to shrink and swell every time it rains and dries up. What do you think it's going to do to your foundation? It's going to rip it apart. Okay? And New York State, you never would have thought that would have occurred because we don't have, well, we do have the right climate, but we're fresher material. You wouldn't think we'd be producing these types of clays, but we in fact do have some of those clays up here. Okay, so here are the orders. Okay, and here are the major characteristics that sort of set that soil apart. Okay? What do we in fact look for to tell us that these are these features? Basically, we look for diagnostic characteristics. Okay, I'm looking for the four legs. I'm looking, well, think of it in evolutionary senses, I'm starting to look for the ear bone. Okay, I'm starting to look for basically mRNA. I'm looking for or hip placement or things like that. I'm looking for does this sound familiar to people? Hip placement, ear bones, stuff like that? Okay, these are diagnostic features of evolutionary history in biological systems. Okay, so I'm going to look at the same thing. I'm going to look at diagnostic features in the evolutionary history of soils. And we have a bunch of them. In the first one, we basically sort of break them apart. We have what we call epipedons, and we have diagnostic subsurface horizons. Epipedons are those diagnostic features we find on the surface of the soil, and the subsurface ones are the ones we find subsurface, down below. Okay? There's a slew of them. Okay? This is a couple of them. They're called epipedons because they're over the pedon. They may include part of the B horizon. Remember, we're talking about horizons versus diagnostic horizons. Okay? There's a variety of them. Some of them are called anthropic. They're human modified. They're high in phosphorus. There's pelagic. There's human modified, but years of manuring. Okay? These are basically what they look like. So we have the molic, we have umbric, ochric, melanic, histic, and we also have, and not on this slide, but we also have this anthropic and the pelagic. Okay, but let's take a look at these five. Okay, these are the epipedons. These are the surface diagnostic features. Okay, we have a molic epipedon. That diagnostic feature that we're looking for is high base saturation, deep high base saturation. Hold on. Okay, does that make sense? Okay, if I have a molic epipedon, the clue is in the name as to what order this is. If I have a molic epipedon, I have a mollisol. Andy?
So can you explain what base saturation is?
Base saturation is a measure, you could have low or high. Base saturation is a measure of how many bases Another way we can sort of think of it at this point, we can think of this as nutrients, as elements. How many bases are present on that soil, or on the particles, or being held by the particles of that soil? That's what base saturation is. So high base saturation is generally a high nutrient soil, but it also could include high salts. So yeah, it would be high nutrients, but potentially it could be problematic. Low base saturation means low nutrients. Okay, does that make sense? Okay, so I have high organic matter, deep, and I have high base saturation. Diagnostic, the characteristic I'm looking for, whether it's the ear bone or whatever, in this case, it's a mollisol. Okay? If I have low base saturation, an umbric epipedon, low base saturation could move into some of those altisols. Okay? If I have an ochric, low base saturation, thin light. Any ideas? Thin light is really the operative term here. Well, potentially. I'm actually more thinking along the lines of insectisols, but it has that low base saturation. So, okay, we move into the melanic. The clue's right there. Melanic, dark, but not organic. Volcanic materials. Next one over, histic. Histosols. Histic, tissue. Okay, the diagnostic feature I'm looking for is organic matter. How much organic matter is in there? Does that make sense? I'm looking for the ear bone, the hip. Go.
The green correlated with the amount of organic material?
No, the green is corresponding to the depth of that diagnostic feature. So mollicks tend to be deep. Umbrics actually tend to be deep as well. Ochric, less. Okay, melanic, someplace in here. Histic, very shallow. Well, it's being deposited on top. Yes, and it does correspond. I mean, ochric does not have a lot of organic matter. This has got the most organic matter, so it's the darkest. Does that sort of answer the question? Go.
So wait, the green from one diagnostic to the other isn't the same in each situation?
The green in this has probably got the most, the green itself is sort of indicative of the organic matter that's in it.
Okay, so with EBOC, the green means organic matter, but with molic, that means...
organic matter as well. No, that means organic matter as well.
Okay, so all green here is just the level of organic matter.
The green represents the intensity of organic matter. I mean, the intensity of the color basically means more accumulation of organic matter. Okay, the bracket basically means how deep this material is, but you can notice in some of these, like the umbric and the mollic, you get down to this end, it's not as dark as up there. But this is the depth of that diagnostic horizon, the epipedon in this case. There's another question, go. You, go.
Okay. The base saturation, does that come from organic matter then?
It could come from organic matter, but it also could come from the weathering. Okay, so in this case, there's not a lot of weathering and not a lot of organic matter. Okay, but I'm starting to see some sort of development in there. Okay, so low base. That doesn't mean that these soils are infertile. It just means it hasn't been weathered. Go.
What are the names represent? What do they represent?
Well, in some cases, this is definitely associated strictly with a mollisol. This is directly associated strictly with an andosol. This is strictly a histosol. But these guys can be multiple soils. Okay, we're going to look at a combination of these diagnostic horizons to tell us what type of soil. I.e., just because I have the ear bone doesn't necessarily mean I'm human. Okay, there's other organisms out there that have the ear bone. I might have the combination of the ear bone, the tooth, and the hip to be said, okay, that's a human. Does that make sense? Okay? All right. So these other diagnostic horizons, we call these diagnostic subsurface horizons. There's more of them. Okay? Soils that have oxic, candic, spodic, argillic, natric, and cambic horizons due to the amount of time they have been developed. These soils, these features are diagnostic due to their development, just like evolution of us. Okay? This is where soil genesis comes back in. Because these features represent differences in that stage of development, in their soil genesis, they are used to group the different soils together. All the soils that have candic horizon, you guys step over here. Everything that has oxic, you're here. Everything that has an argillic, you guys are here. Now that's not to say that there's an overlap on them. We basically are looking at the combinations to figure out what type of species you are or what kind of order you are. Does that make sense? Okay. There are a bunch of them. Okay? I'm going to go through a couple of them. Okay? The first one is an oxic horizon. And an oxic horizon is diagnostic for an oxosoil. In some cases, I just need one diagnostic feature, and that's what it is. If I have a histic epipedon, I've got a histosoil. If I have an oxic horizon, I've got an oxosoil. But that's not the case for all of them. Some of them you have to have the multiples. Okay. If I have a candic or an argillic horizon that has little sodium, potassium, calcium, or magnesium, it's an ultisoil. On the other hand, if I have an argillic or a candic or a natric horizon that has a lot of that stuff, it's an alpha soil. High base saturation, low base saturation. Does that make sense? Sort of that reinforce the high versus low base saturation? Go.
How do you determine if the subsurface horizon is oxic or candic?
A couple ways. There's field techniques that you'll use, but we also send this stuff to the lab. So there's no way that I'm going to be able to take a look at a clay and say it's got low base saturation for high base saturation. High base saturation versus low base saturation.
The subsurface diagnostic horizon it's more like chemical?
Some of it's physical, some of it's chemical. Actually, in some cases, it's biological. Not for the, well, mostly for the ones that are above. For the histic, that's a biological one. I'm looking at accumulation of organic matter. Does that make sense? So what I would do is I would literally cut into the soil, take samples out, take a look at it, and then analyze those samples. Basically the same thing that we would do for organisms. You know, it sounds gruesome, you know, but sometimes we have to cut them open to figure out what's going on. Nowadays, we basically use mRNA. So we'll take a sample, you know, a skin swab or something like that. We'll do a PCR on it so we can amplify the genetics, the RNA, basically, and then we can find out what it is. And we can trace that back, and we can relate that to everything else. It's the same thing that we're doing here. In this case, we're excavating, taking samples out, and analyzing it for certain characteristics. In this case, base saturation. We can figure out the clay content, and then we figure out the base saturation. Okay, and that gives us whether it's an argillic or a candic or whatever it happens to be. Does that make sense? Okay. The next one is a spodec horizon. Now I showed you pictures of these guys, but it's diagnostic. It's a subsurface horizon, diagnostic horizon. It's diagnostic for spodosols. Cambic horizons, diagnostic of inceptisols. Cambic horizon is basically a BW horizon. Okay? Does this sort of start making sense? We're looking for those diagnostic features, i.e. the ear bone. Go.
Didn't you say that high base saturation was like higher nutrients, so you would think that the sodium and the calcium, that would be the higher base saturation?
In this scenario, if I have high potassium, calcium, or magnesium, I'd be here. In this scenario, these are little. They have low. Okay? So if I have low base saturation, we'd call it an ultisoil. If I have high, we would call it an alpha soil. So if you go back to this flow chart, does this flow chart start to make sense? The degree of weathering produces significant features that we can, these significant, well, in some cases, I guess not significant, okay? But we can look at these diagnostic features, combine them, it gives, these features are produced by weathering processes, and that degree of weathering is going to produce the strength or weakness of that diagnostic feature. We put those features together, and we figure out what soils we have. Does that make sense? It literally is exactly what we do with biological systems. We happen to be looking at different characteristics, but okay, so let's look at some of these diagnostic features. And this is out of your book, but this is basically all of the features that we're looking for. This is the ear, the hip, the tooth. This is all these different features, okay, listed. And what we're going to do is we're basically going to break it in half. We'll look at the top half, and then we'll look at the bottom half. Right? So let's start at the top. Here's that recent material. If I have no significant accumulation, okay, and I have a light-colored horizon with some removal, we have an albic horizon. You guys have met albic horizons. What are they called? What is the diagnostic? What's the master horizon term for it? It's an E horizon. Okay, so I look for E horizons, I've got an albic. Okay, on the other hand, if I have weak development, structure, color development, we call it a cambic. What kind of horizon was that? A BW. Okay, so if I start having accumulations of silicate clays, okay, if it's a high activity clay versus a low activity clay, if it's a high activity clay and I have low salt content or low sodium content, it's an argillic. You guys have met this one. This is a BT. On the other hand, if I have low or a high salt content, it's a natric horizon. Clay accumulation but high sodium. On the other hand, I've got low activity clays with moderate weathering and abrupt surface boundaries. It's a candic horizon. If it's under extreme weathering, deep horizons, we call it an oxic. Oxic diagnostic for oxisols. You start seeing the combinations, how you throw these things together, you start getting your soils. All right. If we have an accumulation of sulfur minerals, we've got a sulfuric horizon, diagnostic horizon. If we're having an accumulation of organic matter, cool high altitude tropics, high organic matter, a sombric horizon. Acid weathering with iron and aluminum oxides, a spodec horizon. Clays between a plow layer in cultivated soils, an agric. Keep going. We're just going to go through the varieties here. If I have an accumulation of inorganic salts, and they're carbonates versus gypsum versus a mix. If they're carbonates and they are not cemented, we call it a calcic horizon. If they are cemented, we call it a petrocalcic. If instead of carbonates we're actually looking at gypsum and they're not cemented, we call it a gypsic. If they are cemented, we call it a petrogypsic. Do you guys get the flow? Am I beating a dead horse at this point? You guys will finish it off with the salic? Go. Cemented is when they harden up like a cemented feature versus a soft material. Okay, so if I have accumulations of calcium material and it's not hard, we call it a calcic horizon. On the other hand, if it gets rock-like, we call it a petrocalcic. And I'll show you some pictures of this so you can see the difference. Okay, accumulation of salts, it's mixed, we call it a salic horizon. And I want to talk about these guys as well because you're going to meet this one this week. Okay, if I have cementation of layers that have not anything to do with salts, okay, and they are resistant to water and root penetration, it is cemented due to silica, basically creating a hard pan, we call it a durpan. If it's cemented due to other features, but it's dense and a brittle pan, fragipan. And I'll talk about, this is sort of nebulous at this point. There's a number of different theories as to how this forms, and we'll talk about those theories in the field. On the other hand, if I have a very hard pan that's due to an iron or manganese cementation, we call it a plasic. So we hold these things, we look at all of these in combination, we throw them in together. These are the diagnostic features that tell us what soils orders we're in. Okay? Question. Pan is literally what it sounds like. It's a horizontal sheet of material.
Would it be like the top?
If it's a hard pan, no, it could be in the soil someplace. Okay, so this plastic right here, this is basically formed when the plow goes over something again and again and again. It's a subsurface, very thin. It's a pan, a hard pan. Yeah, some of you guys might have seen the initial formation of it. There was some platey structure right underneath where the AP boundary was with the BW. That was the beginning of this type of pan. It wasn't hard enough to be basically termed a plastic horizon, but that's how they form. Go.
Just judging from that, could you judge which one of the pans it would be? Or would you have to wait till it develops?
In this case, it's weak structural development, so it would actually be a BW, so it would be a cambic.
Okay.
Does that make sense? Because it's not a pan yet. It's not a hard pan yet.
I mean, could you judge which one it would become?
Probably it would come into the... See, this has got iron and manganese cementation, so I'm not sure I'd call it a plasic. It's definitely not going to be a fragipan. It's not. It's. I don't know. I would be stepping off a slippery slope if I started making that. But I hear you, and I'd be like, nah. I'd probably end up going this way, but it's got that iron and manganese cementation, and it's not because of that. This is because we're compacting it. We're hitting it again and again and again. Go.
Can the plow pan cause like any type of pan to form? It just depends on what the soil is?
It has to do with what the soil conditions are. These pans are not forming because of a plow. These pans are forming because of other things. The plow might, in that case, actually might help it happen, but this is not a plow pan. This is a plastic pan. Classic plastic. Okay, does that feel good? Any more questions so far? Okay, so let's take a look. Okay, go.
So is the age of the fan same as Adam's horizons, or is it?
In the case of the arc port, that pan is there only because of management. So those other horizons are definitely older than that pan. Those BTEs EBTs that we saw farther down, those have been going on those have been forming for the last 10, 13,000 years. That pan that we're seeing, that plow pan that we're seeing underneath the AP, that's formed because with the last 100 years. So that's actually kind of interesting because you can see that certain types of processes will produce features very quickly. And some processes will take a long time. It's going to take a long time before that arc port becomes an oxasoil. I mean, a really long time. But if it stays stable, it will form there ultimately, I don't know, 100,000 million years from now. But, okay, you feel comfortable with this? So let's actually take a look at some of what these features look like. Okay, this is a molic epipedon sitting on top of an argillic. You can see how they overlap each other. I got a BT1 and a BT2. I've got an AP, an A2, old BW basically, or E. Actually, I don't know what A2. I don't remember what A2 is. It's the old nomenclature. Okay, but basically we're looking at the molic epipedon overlapping the argillic. Underneath it is a BK. Okay, this is diagnostic K. Carbonates. Okay. But basically what we're looking at here is the two diagnostic features. We put these features together. In this case, the argillic isn't that important for the order classification. It'll be for more refined classification. But we have this molic epipedon. This is probably a mollisol. Does that make sense? Yeah? Okay. All right. So, you've seen this slide before. Okay? This slide is basically parent material. Now, I want to show you this slide. This is a slide that's out of your book. Okay? And they are definitely similar. Okay? There's similarities there if you look at the two of them. Flick it back and forth. This is the map of the soil orders. So these are the 12 soil orders mapped across the United States. Now, that's not to say that everything in here is an ultisoil or everything up here is whatever. Okay? There's a lot of inclusions, but this is the general map of where these orders are. Now, if you look at the parent material, the geology, you can definitely see, if we look at this 7 right in here, we're basically looking at ultisols. Big swath of ultisols right down here. Okay? Now, if we look at this top zone up here, frozen, those are gelsols. We're not going to see the geology on that one because we don't go that far up. But does that make sense? Okay, so we're looking at parent material is not necessarily directly related to what we see on the ground. It does indicate what we're going to see, though. So here's the orders. That's the distribution of them. How do we actually key them out? How do we actually classify them? And we have what's called the keys to soil taxonomy. And what we do is we literally ask specific questions that are diagnostic for that horizon. The very first one is gelsols. Now, starting today and going over the next 2 lectures, we're basically going to go on a safari. We're going to go on a safari and we're going to look at these soils. Now, you've heard me talk about what these are. I want to show you what they are, and then I want to reinforce what we talked about. Say, jello soils. Do you guys remember the diagnostic feature for jello soils are? Frost turning, cryoturbation. Okay? So where are they distributed? Those high elevations, where the frost is, where the permafrost is. This is the United States. We're looking at Alaska. Okay? This is where they are. Okay? The question that we ask, sitting right up there on the top, we're going to ask you one simple question as to what that diagnostic feature is. We're looking for the ear bone or the hip placement. Okay? Soils that have, 1, a permafrost within 1 meter of the soil surface, or gelic materials, that's the cryoturbation materials, within 100 centimeters of the soil surface, and permafrost within 2 meters of the soil surface. Fairly simple, right? The diagnostic feature that I'm looking for is ice. If I have it, I've got a whale. If I have 4 legs and fur and it barks and it licks you every morning, it's a puppy or a dog. Okay, does that make sense? That's what we're looking for here. What do these soils look like? We might actually have to kill another light. Sorry. These are what these soils look like. And this is where we're going to stop today on this rather cool pictures. Okay, lab today, we're going out. We're going to meet across the hall. We're going to meet across the hall for the entire week. And we're going to go out in the field. Make sure you wear your shoes. We're going to expect to have a good time, and we're going to be visiting 2 sites.